Great Ormond Street, wait, I gotta look at my notes. The Great Ormond Street Hospital, the University College of London. And he's gonna talk about the use of amniotic fluid stem cells in treatment of necrotizing enterocolitis. Thank you.
Thank you, Joanne. Thank you for the opportunity to discuss here something that is very close to my heart. In the sense, I'm a pediatric surgeon at Great Ormond Street Hospital, and we face almost on a daily basis with patients that look like this one in the picture. So those are premature babies generally, and they get a terrible disease of the intestine primarily, and they die for a multisystemic organ failure that affect primarily the intestine, but then gets on and destroy their liver, their heart, their brain. Necrotizing enterocolitis doesn't still, unfortunately, have a cure. About 40% of these babies, when they got NEC, they die. As surgeons, we do a very old-fashioned therapy in the sense that we go in, do a laparotomy, and we take out the intestine that is diseased, or make them very sick. So they are left with a short bowel, and if they survive, they are condemned to have TPN and transplantation. So we are really lucky that there's a very reliable model of NEC that is made in pap rats. These are delivered just a bit prematurely, and then they go on to have the same stimuli that those premature babies in human have, in the sense that have formula feeding instead of breastfeeding. So we gavage feed 4 times a day these 4-gram pap rats, and credit should be given to the postdocs doing that. Hypoxia is another stimuli which is very important to create the model, and LPS mimic the bacterial translocation. So in these pap rats, we use stem cells derived from amniotic fluid. We described these cells first with Dr. Tony Asella in Boston at that time, about 8 years ago in the original paper, but essentially we described the possibility of deriving c-KIT positive lineage negative in the amniotic fluid. These cells may have a potential for therapy. Certainly they grow very well outside the body, and they can show different type of engraftment. With Marina Cavazzana-Calvo in Paris, we demonstrate that we can engraft these cells in all hematopoietic lineages in rag minus mice after irradiation, and we have shown that their potential could be just beyond what is recognized of mesenchymal cells, and in fact, we could reprogram these cells to iPS without any genetic manipulation. So by culturing the cells in valproic acid, we can generate truly pluripotent stem cells that are able, when implanting immunodeficient mice, to generate a teratoma. So in these pap rats, we inject cells in the peritoneal cavity. And as you can see here, the numbers of PAPs that we did to validate our results were quite high, about more than 100 PAPs for each arms, and we could see that we could lengthen the survival of these rats. So these rats are not made as a model for long-term survival because you cannot introduce the same maneuver that you would introduce in these babies, such as intubating or giving TPN. It's just not feasible in the 4-gram rats, but we could lengthen survival in a significant way. And in fact, we proved that only the amniotic cells, but no, for example, bone marrow or a lineage of inert myoblast could significantly increase the survival. So when we look at what was happening on those rats, we demonstrate that there was a restore of the motility of the intestine, while the absorption there was a tendency, but that did not reach significant difference. We wanted to have, of course, a parameter to look at that, independent parameter. And so the radiologists look at the MRI of these pap rats, and they could clearly blindly assign the normal pap rats, in which you define a different structure, the one affected by NEC, in which you see fluid inside the lumen of the intestine and outside in the peritoneal cavity, as you commonly see in these babies, also in the clinical model. But most importantly was in the one that were treated with the amniotic fluid stem cells, these features were not recognized. When we look at surgically and sacrifice those animals, clearly you can see in the breastfeed animals there was a normal looking intestine, which was necrotic in the one that had NEC, and was very similar to the normal one in the rats that received the amniotic fluid stem cells. There was some area of inflammation and some probably strictures. The pathology look blindly at those tissues, and you can see disloughing of the mucosa and necrosis of the villi in the animal that got NEC, and the animal that received the cells show a pretty normal restoration of villi anatomy. But when we look at what the cells did, we recognize that only a very few number of cells engraft into the intestine. As you can see here, we could target the cells because they were GFP positive. We could see that they were moving from the mesentery to the wall of the intestine. We now know this probably through a lymphatic way. And these cells are however localized in specific places in the intestine, which are very important for stem cell regulation. So while they move into what it seems the epithelium, but actually is below the epithelium in the axis and in the crypt of the villi, which is very important for the cells and the epithelial restoration. So when we look at how the cells act, because we could not see much direct regeneration, as you observe very few cells engraft, we look, of course, at the microarray first, and we saw, not surprising, that they were involved in proliferation and downregulation of apoptosis and inflammation. This was described before by Stappenberg, who described a very neat model in colon cancer, which is regulated by COX-2 positive cells. So COX-2 positive cells are present in the axis of the villi and move down to the crypt when epithelium needs to grow. Of course, in colonic cancer, there's a dysregulation of this mechanism, and so you have growth and anti-apoptosis lead by the COX-2 positive cells. When we look at the COX-2 positive cells, we recognize quite clearly that in the NEC animals, the COX-2 positive cells were almost disappearing while they were restoring when we were injecting amniotic fluid stem cells. Interesting enough, when we compare breastfeed animals and amniotic fluid stem cells animals with NEC, we observed that there were more cells under the crypt. So when we look at numbers, we could see that there was no significant difference between normal breastfeed animals and animals with NEC that were treated with amniotic fluid stem cells, which is reassuring because of the risk of having high number of COX-2 positive cells can lead to tumor. However, when we look at the position of the cells, we recognize that in the animal that had NEC and had the amniotic fluid stem cells, the cells were mainly present in the crypt, which makes sense because it's where the cells help the epithelium to proliferate. And this is so relevant that when we look at the correlation between the number of COX-2 positive cells and the damage of the epithelium, there was an inverse correlation between the number of the cells in total and the epithelium. And this correlation was even more significant when we look only at the cells present under the crypt. So, of course, you have to randomize your patient, in this case the pup rats, to observe if this is really true. So what we have done was to use breastfeed animals, NEC and NEC with the amniotic fluid stem cells. And then we randomized to 4 subgroups. We randomized to have an inhibitor of COX-2 positive cells, an inhibitor of non-so selective, so COX-1 and COX-2, a COX-1 inhibitor, and just the vehicle that was the DMSO that was given with gavage. So in the survival, and again in the different experiments, you see the curve is exactly the same of what I showed you before. What happened is that if we use COX-1 inhibitor, we did not observe any difference. So the animals were still having the effect of the amniotic fluid stem cells. However, and in support of our hypothesis, when we inhibit the COX-2 pathway, then the effect of the amniotic fluid stem cells was completely abolished. And this doesn't come as a surprise. I'm sure that when we use COX-1, 2 non-selective inhibitor, there was no differences. So our hypothesis is that the cells that we inject through a paracrine mechanism that we are studying at the moment activate the COX-2 positive cells that migrate down into the crypt and activate the epithelium to proliferate and to restore the intestine. We know, for example, that some of those factors are involved. So we observe that when in vitro we stimulate the cells with LPS to mimic what happened in vivo, and then we look at how the cells react to that. We observe a much higher expression of key factors like FGF2 or PDGF-beta were compared, for example, to bone marrow stem cells. So we think that beside thinking that amniotic fluid stem cells can be a source for therapy, we also think that in the necrotizing enterocolitis model, these cells may be therapeutic. Thank you for your attention.
Great, thank you, Paolo. We're going to go through all the talks and then have a panel at the end, so we'll save questions for the end. And I'm going to go next. I'm Joanne Kurtzberg from Duke, and I'm going to talk about using cord blood in regenerative medicine applications in children. If I can have this slide set. Great, thank you. So I need to tell you a little bit about the beginnings of the cord blood story, which was that cord blood was used in 1988 to rescue hematopoiesis in a 5-year-old boy with Fanconi anemia after discovery that his baby sister's cord blood was a match and that his baby sister was not affected with Fanconi anemia. And the transplant was done in France by Alain Gluckman, shown in the white coat in the picture. And fortunately, the transplant was successful. It restored hematopoiesis in the same rate that we would have expected with bone marrow. And the patient is alive and well 28 years later. He's shown in a picture last year with me. And most importantly, his bone marrow and immune system are still durably engrafted with his baby sister's stem cells. We took that work into the clinic, and I don't have time to go into the history, but unrelated cord blood transplant was discovered and validated, and today there are public banks, private banks, and a large source of unrelated cells for transplantation held in various registries. And 40,000 or so unrelated cord blood transplants have been used around the world to treat patients with the same diagnoses that you would treat bone marrow transplant for, like malignancies, hemoglobinopathies, immune deficiency, marrow failure, and certain inherited metabolic diseases. And on our work at Duke, we used cord blood as a donor for hematopoietic stem cell transplantation in patients with inherited metabolic diseases in the categories of leukodystrophies, which are listed on the left hand of the slide. And we observed in that work that the cord blood neurologic outcomes were better than bone marrow transplantation, and over the years have switched to prefer this source for all children, regardless of whether they have a related donor. We also have shown that over time, this treatment affords an 80% event-free survival in these otherwise lethal diseases. But it requires myeloablative chemotherapy, a long stay in the hospital, a risk of graft-versus-host disease, so it's not benign. Just some examples of treatment effects we've observed. This is a child with Hurler syndrome. Untreated, these children die at about 5 years of age. They die of cardiomyopathy, but they also have severe mental retardation. And the curves on the right show the neurologic cognitive development of the first consecutive 20 Hurler patients we transplanted with unrelated donor cord blood, showing in the black dotted line compared to normal children in the red line, that their cognitive function is falling off in the first few years of life, but with treatment, they start to correct and can come back up in the normal range. And I'm old enough now to have followed these patients for a long time. So the two girls in the pictures here are both Hurler patients transplanted when they were about a year of age. They wouldn't be surviving without transplant, but they're both going to their proms. That's their prom picture. But they both have normal intelligence. They're both going to college now. So we know that this cognitive correction is maintained and not just a temporary effect of the treatment. Now, we know in cord blood that there are multiple types of cells, and we know that the hematopoietic stem cells, which is a very small population of the total cord blood cells, are the ones that are responsible for restoring hematopoiesis after myeloablative chemotherapy. But we also in our work have found, and I'm going to show you data about this, that there are other interesting populations of cells in the cord blood, and one that we are focused on is the monocyte population, which contains CD14 cells, which represent roughly 5 to 7% of the cord blood cells in total. And those CD14 cells, when they're isolated as fresh cells, have certain properties and are able to rescue tissue in hypoxic injury models. And we also can manufacture a cell from this CD14 cord blood cells called DUOC, which is effective in inducing remyelination. So with that background, we went into the area of using cord blood without chemotherapy as I think Jan Nolte said yesterday, a living medicine to treat children with various types of brain injury. We started with autologous, or the child's own cord blood, and that required that the family had chosen to bank their baby's cord blood in a private bank when their baby was born. We tested safety and have published results in transfusion in 2010 and found that an intravenous infusion of autologous cord blood in older children was safe. And I'll show you scattered results of studies that we're doing in babies with birth asphyxia or hypoxic ischemic encephalopathy and in cerebral palsy and autism. In addition to that work, we're also working with allogeneic cord blood cells. We're manufacturing a cell we call Duke O cell version 1, or DUOC, and using it to treat children with leukodystrophies, supplementing a standard myeloablative transplant. And then we're looking at unmatched, unrelated cord blood in adults with stroke and sibling cord blood in children with cerebral palsy, trying to address the issue of donor availability to, in the end of the day, have a therapy if this is effective for every patient in need. So first I'll talk about this cell we call DUOC that we manufacture from cord blood. And on the left you can see a picture of a section of the brain of a child who died from Krabbe disease but had received a cord blood transplant with myeloablative chemotherapy 10 months earlier. And in this child, who was a girl, we were able to show with XY FISH that there was engraftment of donor male cells in her brain, and those are the green dot cells with Y chromosomes. And when we looked at what those cells were, they were microglial and oligodendrocytes, and we embarked upon a project to manufacture those cells from both fresh and cryopreserved cord blood, and the cell product you can see swimming around on the slide there is grown in 3 weeks of cell culture with growth factor supplementation. It's an adherent cell, and we can harvest it, and we've created a protocol to treat children intrathecally to supplement a standard transplant. When we studied the function of this cell in various models, we found that it was able to induce remyelination. So this is the Cooperzone model, where you treat mice with Cooperzone, and they demyelinate their corpus callosum. So the top panel shows a controlled mouse that got Cooperzone. And you can see on the left panel, there's very little myelin, which would be blue. In the middle, you can see astrocytosis in the pink, or scarring. And on the right, you can see that by electron microscopy, the rings of myelin are destroyed. Below, we have the same mice treated with Cooperzone, but then rescued with an intracranial injection of the DOOC cells and assessed a week later. And you can see on the left, remyelination by Luxel Fast Blue. In the middle, there's no astrocytosis, and on the right, that the rings of myelin are back to the normal morphology by EM. And that and many other studies we've done have allowed us to see that this cell has the potential to act through a variety of mechanisms, including enzyme replacement, cytokine secretion, which modulates inflammation, drives oligodendrocyte proliferation, and promotes myelination. And we were able then to go into a clinical trial for children with leukodystrophies who also have evidence of corticospinal damage at the time of transplant. And essentially, we take the big portion of the cord blood unit and use that for the systemic transplant. And then a week out, we start the culturing from the small portion of the cord blood unit of the DOOC cells. And 4 weeks post-transplant, we harvest them. And if the child's engrafted systemically without GVH, we give an intrathecal dose of the DOOC cells to augment transplant. So the baby in the picture is a baby with Krabbe disease whose sister died at 10 months of age, but was moribund at 5 months of age. And he is shown in the picture on the bottom left at 5 months of age, doing well. And here he is a year later, year post-transplant, doing well. So we know the transplant has a benefit here, but we also are hoping that we're arresting disease progression earlier with the addition of the DOOC cells. We've treated 9 patients. We have a few more in the pipeline. We have not had safety issues, but we know we can't really assess efficacy in this setting, so we're planning to do manufacturing from the whole cord blood unit and study adult demyelinating diseases without myeloablative chemotherapy. And that's on our docket for the next couple of years. Going back to the fresh CD14s, we've looked at them in various models of hypoxic injury. So the histologic slides on the top show you a model where we can isolate brain tissue ex vivo and propagate it, and then expose it to oxygen glucose deprivation. And if we do that and look at the cells 72 hours later in the middle, you can see that in pink there's a huge astrocytosis and that the green neurons are gone. But if we do the same experiment and add CD14 cells post-shock, you can see on the left that the architecture is preserved, the neurons don't die, and the astrocytosis does not occur. And with that and many other studies, we've gone into studies in the clinic under IND in babies with hypoxic ischemic encephalopathy, giving them back infusions of their own fresh cord blood over the first 2 days of life, and also in adults with ischemic stroke using non-matched allogeneic cord blood. And that one box there is from a paper we published in the Journal of Pediatrics a couple of years ago, showing with a non-randomized concomitant control group that if babies with HIE got cooled and also received cells, they had better one-year outcomes, meaning better survival with normal function than babies who got cooled alone. And based on all of this data, we've been able to go into the clinic now and randomize phase 2 studies in HIE, which is a multi-center study, and also adult stroke. So maybe next year I can tell you more about that. The other thing we focused on is looking at the effect of autologous cord blood in treating children with cerebral palsy, particularly spastic cerebral palsy due to stroke or HIE or PVL or prematurity. at birth. And over the past several years, we treated 63 patients on a randomized, placebo-controlled, blinded trial of autologous cord blood, where children received a single IV infusion in the outpatient clinic after pre-medication and were followed with multiple testing. The placebo that we used was tissue culture media with DMSO, so everybody got pink stuff and everybody smelled. And I'll show you the scheme in a minute. And our primary endpoint was change from expected GMFM score, which is a motor function score, at 1 and 2 years. So this was our scheme. Children were qualified, their cord blood was qualified, they were enrolled and randomized, and they either received cord blood or placebo, had a bunch of tests done, came back a year later, which was our primary evaluation, and crossed over to get placebo or cord blood, whichever they didn't get the first time, all blinded, and then they were evaluated at year 2. And the primary endpoint was based on this gross motor function measure, where 66 different points are evaluated by an observer of the child at each time point. And children can be graded by their severity and then also by age. So in this score, different children will have different responses depending on when they come onto the study by age and level. And to assess a change, we had to look at where the child was, where they were predicted to be, and then where they really were and how different that was from where they were predicted to be. So you can't just use an absolute number, because a young kid with a mild form of CP will be expected by natural history to have a lot of change. And that wouldn't be a response to the therapy. So we did all those analyses, and long story short, we were able to show that children, and these are 1-year assessments, that the children who received either a higher dose of cord blood cells, which was 25 million cells per kilo or greater, in green, had a statistically significant improvement in motor function at a year compared to either placebo in blue or low-dose cells in red. And we're confirming that with 2-year follow-up now, but the data is looking good. We also did MRI surrogate or biomarker studies, and the top panel shows you the differential increase in connectivity in the low-dose group on the left or the high-dose group on the right. And again, there was a statistically significant increase in connectivity in the high-dose group. And you can see it in the picture. So the top picture is baseline in 1 year of a child who had a high change score and a high clinical improvement and a high dose. And if you look at the red, you can see that at 1 year it's thicker. And on the bottom, you can see that an example of a child who did not improve and did not get cells and had actually more tissue in the red dye at 1 year, at baseline, sorry, than 1 year. So this is a good correlate, we think, with clinical outcomes. Our conclusion from all of that was that cord blood was feasible to give as an autologous infusion in kids with CP, and that kids getting an appropriate dose had improvement. And I didn't show you the data, but this was not impacted by age or severity of CP within the group, which was 1 to 6 years of age when they came on study. But we know that since a lot of children don't have their own cord blood but could have cerebral palsy, we felt we had to look and see if an allogeneic product would have similar results. And so our current work is focusing on that. And we've done a safety study in 15 children getting sibling cord blood. And we're planning to open this winter or spring a phase 2 randomized trial of allogeneic cord blood in children with CP who don't have their own cells. Finally, I just want to show you some results of a trial we're doing in children with autism. Again, we started with autologous cells, and in this trial, they had to have a qualified cord blood, and our hypothesis was that the cord blood cells would secrete factors that would modulate inflammation and suppress microglial activation in these kids. We performed an open-label study in 25 children, ages 2 to 6, and we used the study to evaluate both safety and also define endpoints for a phase 2 study. We developed an in vitro assay to look at microglial activation. So normal microglia are on the left. When they're exposed to LPC, which will induce inflammation, they get microglial activation on the upper right. And when we put cord blood cells in on the bottom, you can see that that microglial activation is suppressed. And we plan to use that as a potency assay going forward. So in the phase 1 study, we did not notice any serious adverse events and felt it was tolerable. And we defined endpoints that we think are going to be useful in a phase 2 study. And we did describe clinical outcomes at 6 months and a year. And we did see improvement in more than half of the children on the study in a number of the subscales of different tests that are used to assess behavior in children with autistic features. And these are now the endpoints we chose for the next study. This is the Vineland, and I just want to show you that when we looked at the Vineland overall in the patients on the lower right, we did see at 6 months statistically significant increase in function in the overall population or improvement in behavior in the overall population, which was sustained but did not further improve at 12 months. But when we looked on the bottom right at which children were responding, the children with the nonverbal IQ of 70 or higher in red had a better response than children with a lower nonverbal IQ. And again, we're using this modeling to go into a phase 2 randomized trial, which is now open, will enroll close to 200 children, has enrolled about 40 so far. But this is what we call a best donors trial. So children are evaluated, their donor is evaluated, if they're eligible, they're randomized, and it's a 2-to-1 randomization between cells and placebo. And if children have autologous cells that qualify, they get them. If not, they get unrelated donor cells. We're not using sibling cells in this trial in case there's some genetic component to autism. And the children are screened to not qualify any children that have known mutations associated with autism, ASD syndrome diseases. So in summary, cord blood therapies show promising results for treatment of diseases with no currently available therapy. Results using autologous cells in babies and children with HIE, CP, and autism suggest efficacy. Safety of allogeneic cells has been demonstrated, and we're moving forward with efficacy studies of allogeneic and best donor cells. Don't have time to talk about it today, but regulatory pathways for both of these types of therapies need to be further defined to really bring this to the clinic. And I'll just end by thanking my whole team. It takes hundreds of people in all sorts of disciplines to really get these studies done, and it really takes a village. And we have support from multiple foundations, which has really enabled the clinical trials to be done, specifically the Julian Robertson Foundation, the Marcus Foundation, the Cure CP Foundation. And the 3 kids in the picture are kids we took to the FDA to help get the indication for allogeneic cord blood, unrelated cord blood, to be expanded to allow for treatment of kids with metabolic diseases. The little girl has, on the bottom with the blue shirt, has Krabbe disease, transplanted at 19 days of age with unrelated cord blood. The girl in the black sweater is one of the kids at the prom with Hurler syndrome. And the little boy in the blue check shirt is a kid with adrenal leukodystrophy. And they all, I think, were instrumental in convincing the FDA to use the right language in the indication to enable the therapy. So thank you very much. So it's my pleasure now to introduce Julie Alexson, who's director of the Wake Forest Regenerative Medicine Center at the Wake Forest Institute of Regenerative Medicine. I may not have said that exactly right. And Julie is going to talk about amniotic fluid cells, banking, and their manufacturing for use in the clinic. Thank you.
Good afternoon. Thank you. I'd like to thank the organizers and Dr. Kurtzberg and the Cord Blood Association who sponsored this session today. So I'm really excited. There's a lot of efforts moving forward in perinatal tissue. So I wanted to be able to touch on that and talk a little bit about some of the banking that we're doing at Wake Forest. So we have about 450 faculty and staff that make up the Institute of Regenerative Medicine. That's led by Dr. Tony Atala. And I really have to show the mission before I start because that's what drives everybody. It's this incredible energy that you feel when you get into the Institute. So we've had a lot of firsts in the field, but one of the things that we've worked on is stem cells from placenta and amniotic fluid, which Dr. Jacoby spoke about the amniotic fluid. So this is about 15 years of an effort so far, working with the cells in preclinical studies and looking at process development and moving them into a clinical master cell bank. So we know these cells can be collected at any time during development and then postnatal. So there's a lot of opportunities to collect these cells at 10 to 12 weeks gestation in a source of amniotic fluid or CVS, and then at term in placenta or amniotic fluid or the amniotic membrane. So these are just a few of the different sources that are used and that are actually being looked at in preclinical studies as well as being moved to clinic. So this is the paper that was led by Dr. Jacoby, a pivotal paper in 2007 that described the amniotic fluid and the placental stem cells and their plasticity and all the work that had been done, looking at the fact that these cells are non-tumorigenic. So they're close to an embryonic stem cell, but they're in between, I guess, what I would say bone marrow and embryonic stem cell, but closer to the embryonic stem cell. So this is the profile, which is basically the same as a mesenchymal stem cell, except you have a strong SSEA4 and OCT4 that's presented. And when we look at these cells, if we look at the cells from the 10 to 12 weeks gestation, we see that we can take them out over 250 population doublings, and we see that they maintain their telomere length from 20 to 250 population doublings. You see that in line 3 and 4 of the telomere length. It retains a normal DNA content as well as the karyotype. So these cells are pretty hardy, they're very plastic, they can differentiate into the 3 different germ cell layers, and they actually have immune modulation capacity. So as we see, these cells can inhibit lymphocyte activation and producing factors that can regulate the immune response. And as we compare those to bone marrow mesenchymal cells, we see a few additional factors that they produce, which may produce a little bit of a different effect than the mesenchymal stem cells. This is just an example of some of the tissue engineering that we looked at with bone differentiation. So taking the amniotic fluid stem cells, differentiating them to osteogenic cells, and then seeding them into the scaffold and implanting those into a mouse model. On the left, you see the scaffold alone that we took out by a Vancosa stain, and then next to that is the amniotic fluid where you're seeing the dark staining, so the mineralization from the calcium. Next to that is a micro CT scan of 18 weeks after implant, and you see on the left the scaffold, or the right, I'm sorry, was just the scaffold without the cells, and then on the left is the construct that was implanted, and you can see that out 18 weeks. So the cells have been used functionally and looked at functionally in several different animal models, and this one is just demonstrating the capacity to produce neonatal tissue, and they've been able to differentiate into podocytes, so they are compatible with the kidney tissue. And this is a few of the recent publications, but I'll tell you there's probably 100+ publications looking at the cell characterization as well as the functionality of the cell. And I guess I'd just call your attention to 2 of the studies. One is in osteogenic imperfecta, where the cells were able to differentiate and amend the brittle bone disease as we looked at those in an animal model, as well as Allport syndrome. So again, it was able to differentiate into podocytes and demonstrate efficacy, decrease the blood urea in the animal, and inflammation as well. So I went back to look at the clinical trials, so how many cells have gone into clinical trials, and did a search on looking at placental cell therapy. I found about 82, and I'm sure that, maybe they weren't all placental, but there were a lot of them. I noted some of the applications that we see a lot for diabetic neuropathies and critical limb ischemia. Amniotic cell therapy, about half that you see, which includes amniotic membrane, a whole membrane, a decellularized membrane, and the amniotic fluid stem cells. and umbilical cord tissue, which came along a little bit later, maybe about 5 years ago, where people really started looking at privately banking that and then started looking at the efficacy of those cells. So at Wake Forest, we bank placental cells. We set up a collection kit that goes over to the hospital. They collect the placenta either at a C-section or vaginal birth. That comes back to our facility, and then we decontaminate that organ. We biopsy the section of the tissue that's appropriate. We digest that tissue, and then we expand those cells. The selection of the tissue is very important because you have not only the fetal but the maternal cells that are present in the tissue. So it is a challenge. You need to be able to get to the right space in the tissue to be able to collect those cells. Otherwise, you'd have maternal cell contamination. If you get a certain amount of contamination, maybe even close to 30%, those can outrun your fetal cells and you could end up with maternal cells. So it is really important to be able to characterize and determine what your cell population is at the end. So just to go over, these are the characteristics. Mesenchymal stem cell with the SSCA4 and OCT4 expression. They double about every 36 hours. They can maintain their telomere length. These clonal cell lines that were produced go out over 250 population doublings. We see no evidence of tumor genicity and really the capability of being able to differentiate into cells from the 3 germ cell layers. So we produce these master cell banks in our GMP facility. It's about 4,000 square feet. We work on tissue engineering organs, cell therapy, biomaterials, and small molecules in this facility. And we're really set up for innovation and really organized for translation. We have these core facilities that specialize in the area. They do training. They might do the testing for us, and they're staffed. So that really helps to speed along the translation as the product moves into preclinical development through process development and manufacturing. We're right now setting up a whole GLP facility that simulates a clean room so that we can make our preclinical test model articles in there as well as our process development. So we're not mixing anything in the manufacturing facility. It's all separated. This is our So I won't go through all this, but these are the steps, the process map as we're producing the master cell bank and all the testing that has to go on, which is really according to FDA criteria. And just want to say that there's a lot of opportunities as you're taking these cells and products to clinic. So right now with this product, as we're making master cell banks, until we really find a clinical application, those cells won't be required to be registered or an IND. Once we determine the clinical application, then we'll put in an IND. So right now the FDA doesn't really want to look at the cells as they're going into a master cell bank. Certainly you could tap on them for questions, but as you're moving to a cell therapy, you have a lot of opportunity to speak to them. So I guess I would really encourage you to go in with a strong plan and then get their feedback, because you are the experts at your technology, but you definitely want their feedback. You don't want to have to go back and repeat testing after you've gone through all the effort and costs. So I guess in summary, I just want to say that birthing tissues are definitely maturing. We're seeing a lot more collected, stored as private banks, and we think there are a lot of opportunities to be able to use these cells as an off-the-shelf product. They have potential as they're plastic. They can be controlled, so better than an embryonic stem cell, and the fact that there's no teratomas. There's people using the cells, the tissues, the exosomes. There's a lot of different products from these cells and tissues. And again, I would recommend that you consult your regulatory authority to be able to speed along to your translation to commercialization. Thank you.
And our last speaker is Todd McAllister, and he is director of the Amnion Foundation, which is a public stem cell bank. And he's going to tell us a little bit about it.
Thanks, Joanne. We wanted to finish the session with just a few minutes to remind everybody of the role that public stem cell banks play in this development. And I think as we've seen all of these technologies are starting to march towards the clinic, but we're still in relatively small numbers. So the question, of course, is how do we translate all of this from small, limited clinical studies to widespread clinical use? And just as a very brief overview for the session in general and some of the things that we've seen on the other days of this meeting, I think it's quite clear that there's not only expanding indications for cord blood derived cells, but also an expanded source of cells. So we've talked about mesenchymal stem cells from umbilical cord tissue. Paolo and Julie also talked about the amniotic derived stem cells. And again, while it's been very promising work, these are still relatively limited and generally autologous studies. And the challenge, of course, and I think Joanne would probably be the first to tell you, it takes years to enroll these patients in relatively small numbers if we're limited exclusively to the autologous model. So all of our goals, I think, is to be able to deliver this to widespread clinical use, and in general that means an allogeneic model. So the public banking model, I think, is very well established for cord blood, but not so well established at this point for these newer tissues and these newer sources that are coming along. So whether that's umbilical tissue or, again, amniotic-derived cells, that model is not yet into clinical practice. But I think the timing is appropriate. Again, Paolo and Julie both spoke to the fact that the amniotic-derived stem cells have now got more than a decade of developmental work in the clinic, and we're marching towards at least limited clinical use. So in parallel with those academic efforts, we've started to build a public stem cell bank located also in Winston-Salem, in fact, just across the street from Wake Forest. And our objective, as I'll show you in the next slide, is to be able to build a large public bank of these immunologically matched cells available to treat patients for these broader therapeutic applications. I won't go into detail on the science of this. I think you've heard that now a couple of times. But again, I think the key point here is we've got a very strong academic foundation for these cells as well as other cell types, and we're interested in being able to exploit that on a wider spread basis. So what is Amnion intending to do? Well, we're a nonprofit stem cell bank, as I said, located in Winston-Salem, and we really have three primary objectives. The first and most obvious is simply to put together the stem cell bank and build a bank of tissues to serve a large population. The second, and maybe a little bit less obvious, but we're also trying to support academic research using cord blood or placenta-derived stem cells. So we are philanthropically supported, but part of our mission is to partner with the best and the brightest from the academic community and support those studies. And finally, and this is something that's not often talked about, but again, I'm sure Joanne can talk to this point, most of the patients really have no understanding or no clue of the power of these cells, the risks, the benefits, et cetera. And while there are now a variety of different patient advocacy groups out there, one of our principal missions is to be able to provide educational tools and outreach tools for these patients to really truly understand what the risks and benefits are for stem cell therapies. And so I'll finish my little blurb here on Amnion by simply saying we are actively now pursuing partnerships and collaborative opportunities. That could be partnering to accelerate our tissue collection. It could also be grants and partnering opportunities that we're sponsoring and supporting. And of course, as I just mentioned in the last slide, trying to partner around patient education and patient advocacy. So with that, I'll finish my little 3 or 4 minute discussion on the impact and the importance of public stem cell banking, and encourage you to look at our website to learn a little bit more about what we'll be building over the coming years. Thank you.
So since we got started late, I want to give you, the audience, the opportunity to ask any of the speakers a question before we chat ourselves. Can we have the microphones on, please?
Thank you. Actually, I don't want to destroy the party here, but I have major fundamental questions here. I heard the word stem cells about 1,000 times in this conversation here, and I wonder, are these really stem cells? They were defined as something between embryonic to adult stem cells, while it's questionable whether adult stem cells are stem cells. And I really tried to get point by point. As to the placental cells, the stromal placental cells have been shown by various researchers are not stem cells, are very potent cells. They have indirect effect on regeneration. They induce stem cells proliferation, but they by themselves have no stemness. So the stemness is questionable all along this discussion that we had here. And I'll go point by point. When we look, for instance, at the gut, and we see few cells that are integrating within one crypt, I look at it and physiologically understand they don't act as stem cells there, because every crypt is formed by single stem cells. So if there were stem cells, I would expect to see a whole crypt which is formed by these cells and not integrated in. I don't say that they don't help regenerate the tissue, but they are secretors that help the body regenerate itself. And it has been discussed in various forums here, especially by Kaplan, who actually formed the terminology mesenchymal stem cells, and now admits that he actually was wrong. These are pharmaceutical factories rather than being stem cells that are involved in building tissues as building blocks, as one would expect. So it seems very sexy and nice, but actually it hasn't been proved. Thank you.
So we're going to have a two-pronged answer here. I will challenge you and say that in cord blood there are blood stem cells that engraft and repopulate and reconstitute hematopoiesis for decades, and we have evidence of that. But there are other cells that are effector cells, you're right, and are not stem cells. Why don't you guys address the amniotic cells?
Yeah, I mean, on the amniotic cells as well, we demonstrated a similar way in the sense that, for example, for the matter of time, I just show, because they are published data, but we've shown that in the RAG minus mice, after irradiation, we can repopulate at first, secondary, and tertiary transplantation. So they behave as stem cells. In other example, we have seen, like in the intestine, as you say, that they have a paracrine action. I think we have to agree on the nomenclature in the sense that if we consider stem cells that are like embryonic stem cells, clearly these are not stem cells. But however, stem cells have different potential, and I would define them as broadly multipotent, as we demonstrated in other experiments.
Yeah, I would echo what Paulo said, and I am cautious to say multipotent, so I think that there's different actions depending on the therapeutic application.
Okay, we'll take one more question because we have to stay on time. Yeah.
Hi, I'm Victoria Nicholas from Proactive Bioscience and from UCLA. This question is for Dr. DeCoppi. So as you know, breast milk feeding is the one intervention in preterm infants that actually reduces and nearly eliminates necrotizing enterocolitis. So I wonder if in your model system where the amniotic fluid stem cells actually repopulate the COX-2 positive cells, so is the effect in the breastfed model, is that due to a stem cell from breast milk, which is known to exist, or is that a factor in breast milk that supports that population of cells, the COX-2 positive cells in particular?
This is a very, very interesting question. The breastfeed, unfortunately, reduces the incidence of NEC, but doesn't eliminate, in the sense that you have breastfeed term babies that get NEC, unfortunately. That type of experiment, unfortunately, has not been done yet, and I think it should be done. Clearly, there are some progenitors, let's use this term, in the breastfeed that may regulate the intestine and actually may be very relevant to the NEC. Other factors, though, may contribute as well in the breastfeed. You have antibodies in the breastfeed, you have immunological cells that can help as well. So the breastfeed is certainly a very interesting source, is undervaluated at the moment and is very important, certainly in a newborn. Yeah.
Okay. Well, I want to thank our speakers and thank everyone for attending. Sorry we got started a little late, but we're going to stay on time now. The speakers are here, and if you have other questions for them, you can come up and we can talk individually. Thank you very much.